I'm going to talk about the dynamics of human cytomegalovirus infection during differentiation of CD34 positive progenitor cells into dendritic cells. So human cytomegalovirus is a beta herpes virus, and although it does not cause disease in immunocompetent hosts, it's a major cause of morbidity and mortality in immunocompromised individuals and congenitally infected babies. CMV is able to infect a wide variety of cells, including CD34 positive hematopoietic stem cells and dendritic cells. So as all herpes viruses, cytomegalovirus establishes latency, a lifelong latency after a primary infection. So in the case of CMV, the primary infection happens when there is contact of the oral or nasal mucosa with contaminated saliva or urine. The virus then replicates in the mucosa and is spread through the blood, getting to the bone marrow. In the bone marrow, those viruses will infect CD34 positive cells and will establish a lifelong latency in those cells. During this period of latency, there is no production of infectious particles. From time to time, those viruses are able to reactivate, and then there is production of infectious viral particles. They are disseminated via bodily fluids like urine and saliva. For the purpose of this talk, we are going to focus on the latency and reactivation part of CMV pathogenesis. So as I said before, the hematopoietic stem cells are one of the sites where CMV is able to establish latency. Those cells are going to originate all the cells in the blood. So any latent genome that is present in the hematopoietic cells are going to be carried over to differentiated cells, including dendritic cells. The dendritic cells are very important for CMV pathogenesis, mainly a subtype that is present in the oral mucosa called Langerhans type cells. They are sites of reactivation of the virus, so they might contribute to viral dissemination by releasing reactivated virus in the saliva. So in order to understand CMV pathogenesis, it is important to know what are the relationships with the relationships of the virus with hematopoietic stem cells and dendritic cells. So our main goal in this project is to see what happens when we infect CD34 cells and those viruses go latent, what happens with the virus during the path to differentiation of those cells in immature LCs, and the maturation of those cells. The specific goal, one of the specific goals is to quantify both cellular and viral genomes during this process. The percentage of the cells that are able to express immediate early proteins, which means that the virus are entering the lytic cycle instead of staying latent, and to quantify the viral progeny production, again, to see if the virus is going latent or going lytic or staying latent. And this is the first work that actually quantifies what happens during this path. The second goal is to compare two strains, TB40BAC4 and AG169. TB40BAC4 is a clinical-like strain, so it's similar to the CMV that is circulating in the population, and it has an intact genome, so there are no genes missing from this strain. On the other hand, AG169 is an attenuated virus, it's a vaccine strain, and has been extensively passed in fibroblasts. During these passages, this strain lost a number of genes, including genes that are important for latency establishment. What is interesting about this strain is also that there is a controversy about the ability of this strain to establish latency in CD34. So there are groups that say if you infect CD34 with AG169, you actually have the AG69 going lytic instead of latent, while other groups show that no, the AG69 goes latent in CD34 cells. So we wanted to know what's going to happen in our system. And the last goal is to see, to assess the role of maturation of IC in virus reactivation. In the literature, maturation with LPS has been linked to virus reactivation. So the way you're going to do this, we're going to infect CD34 cells with both strains, incubate those cells in differentiation medium, and so they will differentiate into immature LCs. And we are going to collect points during those 8 days. After 8 days, those cells will be harvested, and they are replaced in either differentiation medium, so they keep as immature cells, or they are mature with the addition of CD40 ligand and LPS. So here are the results of qPCR assays, and we are going to focus on the first part, that is the differentiation of CD34 into ILCs. We can see that the cells infected with TB40, they are able to proliferate, so in green are the genome cells. When you look at the viral genomes, you see a decrease in the number of genomes up to day 4, and then this number stabilizes in around 20% of the initial input. With AG169, we see that the cells are again able to proliferate, but you see a more drastic decrease in the number of viral genomes. This decrease, though, is much less if you start the infection with 10 times higher the initial input than in this situation. Then you have a stabilization of the number of genomes in around 20 to 36% of the initial input. So what this tells us is that both viruses are able to maintain their genomes in those cells while they are differentiating, but AG169 is less efficient than TB40, BAC4. In none of the time points, we saw expression of immediate early proteins or viral progeny production, which means those viruses are establishing non-lytic infections in the CD34. They're differentiating into ILCs. And it also tells us that none of the genes missing from the AG169 genome, in particular UL138, that has been linked to latency establishment, none of those genes appear to be important for AG169 to establish latence in these cells. Now moving forward to if those cells are kept as immature cells or if they are matured, we can see right after the replating of those cells, there is a cell loss, but those cells recuperate and they start proliferating for both ILCs and MLCs. When you look at the viral genomes, you also see a drop in the number of viral genomes. There is probably associated with the cell loss in the beginning of the experiment. But you see that there is genome maintenance for TB40 in both ILC and MLC. The difference is that MLC seems more, the efficiency of genome maintenance in MLC is higher than in ILC for TB40. With AG169, we see the same trend for the cells, and we see again a more drastic genome loss with the AG169. If you start the infection with 10 times higher input, you have more genomes at the end being maintained, but on the contrary of TB40, you don't have a difference in genome maintenance between immature and mature LC. Again, we didn't see expression of immediate early proteins or viral progeny production in both immature and mature. And actually, the mature was a little surprising because, as I said before, maturation was linked to virus reactivation, and that's not what we observed in this case. So as the main conclusions of the work is that both strains are able to maintain their genomes during the path of differentiation and maturation of CD34. This genome maintenance is done in the absence of immediate early gene expression and in the absence of viral particle production. And it's important to point that even AG169 was able to establish non-lytic infection However, it's less efficient in maintaining their genomes in those cells than TB40. The second important conclusion was that the maturation using CD40 ligand and LPS was not able to induce virus reactivation. So what we think is that additional stimulation is necessary for the virus to reactivate. And the future directions for this work are to assess the behavior of other CMV strains that lack specific proteins that are involved in both virotropism and latent establishment, assess the behavior of CMV in CD34 cells that are maintained in conditions of self-renewal instead of differentiation, and to test the effects of different maturation stimuli on viral reactivation and see if we can get this reactivation. I would like to thank the people from the lab, past and present, my PI, Dr. Laura Hertel, and the funding sources. Thank you.
Very nice talk.
Thank you.
I think that CMV reactivation is a big problem after bone marrow transplantation, for example, in leukemia patients.
Yes.
So do you think the CMV gets reactivated from the recipient, from non-hematopoietic cells, because all the CD34 or stem cells of the recipient should be eliminated? Or do you think it's being reactivated from the stem cells, hematopoietic stem cells from the donor?
Usually it reactivates from the donor. Mm-hmm. And it's a bigger problem if the recipient never had contact with CMV. It's also a problem because the transplant people are immunosuppressed. Yeah. And then CMV is able to start replicating, while if you're immunocompetent, the immune system is able to keep the CMV at bay. So we can't eliminate the CD34 cells because they are the ones that are going to be responsible to actually do the job of making the new cells. They're not cancerous. So the thing is to come up with vaccines or antivirus, and people do use antivirus in the clinic to try to inhibit CMV replication in those cases.
Yeah. Okay. Thank you.
Hi, it's a very nice presentation. Your system
Thank
is
you.
quite exquisite. What's interesting is you're not getting detection of any of the immediate early proteins in your system. Mm-hmm. Earlier work with Greg Perry has indicated that IE1 expression during latency seems to contribute to the genome maintenance. Are there differences between your system and theirs that might explain your different results?
So what is different from our system is that our system is more defined. So we don't use serum in the media, and we know exactly which cytokines we add to those cells. So the CD34 cells are very sensitive to any type of stimuli. So if you don't know exactly what you're putting in the media, you actually can't tell what exactly they are differentiating into. So that's one important thing, one important difference about our system and other systems.
Can you enhance add 169 DNA genome stability if you infect cells with both viruses?
We never try to do that. I would say it might be possible because because any of the genes that are missing on AG169, then the other virus would provide it, but we've never done this.
Okay, last question. If you had to guess what would be the stimulant thing for reactivation, because you're using the one that are already sought to reactivate, where you put in a catadine, you get all the...
Yeah, so
Yeah, I was just wondering about the stimuli that she thinks are missing to reactivate, because, you know, the main ones are, you know, activated by CD40. I mean, the one that are thought to be activated at least by LPS.
So we actually have a project going on in the lab using different cytokines. The results are still preliminary, and Roxanne is actually going to talk about this a little bit during the poster section.
No?
But usually in CMV field, what people do for reactivating those cells is incubating the CD34 cells with fibroblasts. And then we have no idea what types of signals the fibroblasts are actually giving to the CD34, so the virus can reactivate. But
Okay, thank you.